This video covers the IGCSE Biology Objective 4.1.3b on the 2025 CAIE syllabus to describe the use of Benedict's solution test for reducing sugars. So Benedict's solution is pictured here. It's an indicator for reducing sugars, which we'll talk about what reducing sugars are in just a minute. But just to remind ourselves, an indicator is a chemical which changes color in the presence of another specific chemical. So Benedict's solution, the indicator, original color is blue. In the presence of a reducing sugar, which is the specific chemical, it would then change color. And we'll see what those colors are in just a minute. Again, hopefully you'll have an opportunity to do this particular practical in the lab, but if you don't, then you have the information here. So what are some reducing sugars? Reducing means that it's going to donate electrons to another chemical. The chemical that it donates to is the Benedict solution, and that's what now is going to cause the color change in the Benedict solution. So if you recall, we have three categories of carbohydrates. There are monosaccharides, disaccharides, and polysaccharides. Now we have already determined that iodine is going to be the indicator test for starch. So now we're looking at the monosaccharides and disaccharides. So all of our monosaccharides, that is glucose, fructose, and galactose, are all reducing sugars. They all donate electrons to the Benedict solution, and therefore the Benedict solution will change color. For the disaccharide molecules, it is lactose and maltose that will donate electrons to the Benedict solution, and therefore it would turn another color besides the blue. Sucrose, which is the third disaccharide, and remember sucrose is table sugar, does not donate electrons, so it is not considered a reducing sugar, and so you would have to do a different type of test to determine if sucrose was in the food molecule, the food sample that you were testing. So for the most part, all of the monosaccharides and 2 out of the 3 disaccharides are all reducing sugars. So the Benedict's color change. So what you do is you expose the solution in question, or the food in question, dissolved food, in water, and then you would expose it to the Benedict solution. You would just put some Benedict solution into the test tube, and then you would leave it in a hot water bath for about 5 to 7 minutes. And basically what you're going to have, if you have a positive result, if you have a reducing sugar, any one of the 5 that I previously mentioned, you would have a range of color changes from green all the way to red. And so you can see there on the right hand side a picture of an actual test with a different amounts of reducing sugar. So if your Benedict color change stopped at green, that means there's only trace amounts of reducing sugar in the food sample that you are working with. If it stopped at yellow, it would have slightly more, but not a lot, of reducing sugar in the sample. And then you would go to orange and then to red. The red, what's called the brick red, has the highest amount of reducing sugar. This would be a sample of like pure glucose. So if you had glucose powder and you dissolved it in water and then did this Benedict solution test where you poured Benedict solution into it, now it would turn blue, and then you put it in a hot water bath for 5 to 7 minutes, it would go through all of these color changes. It would go green and then yellow and then orange and then brick red to show you that particular color range.